And regarding cell cycle, these are the books I've referred, so you have an idea of most of the books. When we come to this concept of cell cycle, since you have studied cell cycle in your class 11 and 12, you must have studied that every cell has a life. Just like we have a life, a cell has a cellular life. So in its cellular life, it passes through a cycle in a cyclical manner as long as the uh, DNA of the cell allows it to permits it to pass in that cycle. Because if you remember, during replication, we had studied this concept of end replication problem. So there was this point that after end of every cell cycle, that means after end of every replication that is occurring within the cell cycle, the length of the DNA at the ends is getting shortened. And this goes on happening. The DNA gets shortened again and again with the preceding amounts of cell cycle, and a time comes when the DNA reaches a critical length. It is then that the cell is not allowed to pass anymore through the cell cycle, and then the cell is instructed to die a apoptotic death. We will come to the death part later. We will concentrate ourselves on cell cycle. So you people are all aware the cell passes through, sorry, the cell has a G1 phase. So you're all aware of the fact the cell has a G1 phase. So let's start with G1. This is the point where RNA and protein synthesis occurs. No DNA synthesis occurs here. In case of a standard human cell, this is about 6 to 12 hours. So the cell is preparing for a very vital task, the task of replication that is about to start in the S phase. The name S comes from the word synthesis. So this is the synthesis phase for DNA replication. The name G comes from gap. So this is gap 1. Cell is not as such doing any function that can be seen from outside, but it is a very vital stage where the cell is preparing itself. The cell has just come out of mitosis. The cell will enter the stage of DNA replication. So coming out of mitosis and entering the DNA replication stage, the cell needs to prepare itself with all those proteins, RNAs that are required for DNA replication. So even though externally we do not find much changes in the cell going on, but at the cellular level, gap 1 is a very important stage. And as I said, in humans, it is about 6 to 12 hours. And as the cell is prepared, when the completion of preparation of the cell has taken place, the cell enters the S phase. In S phase, we have DNA replication. DNA doubles, RNA and proteins are also synthesized. If we look into a traditional human cell cycle hours, this is 6 to 8 hours. After the replication has completed, there is again a small gap 2 phase where the cell comes out from DNA replication, but the cell is now preparing itself for mitosis, where you know that cellular division will take place. Karyokinesis followed by cytokinesis will occur. So cell again needs to prepare itself for the mitosis stage. So after coming out of this synthesis stage where DNA replication has occurred, the cell enters the gap 2 phase, which is about 3 to 4 hours. No DNA synthesis occurs here. RNA and protein synthesis continues. And finally, the cell enters the stage of mitosis. You have all studied mitosis in details. This roughly takes place for 1 hour. So as you can see, with respect to the time, the maximum hours that the cell is spending is in gap 1 phase. Because gap 1 is a very critical phase, and we'll understand how critical gap 1 is. So this is only for 1 hour, where mitosis, that is nuclear division, and cytokinesis, that is cell division, ultimately yields two daughter cells. So this is a life of a cell from mitosis gap 1 S, gap 2 mitosis. Just remember, if a cell is entering gap 1 with 2n number of chromosomes, so when it is entering the S phase, the chromosome number has doubled, isn't it? Because DNA replication has taken place. In gap 2, it's again doubled chromosome DNA amount. It's the double DNA content. And finally, when it enters mitosis, we get the division of this doubled DNA content into two daughter cells. So from the DNA point of view, in gap 1, we are having 2n amount of DNA, that is the amount of DNA present in a diploid set. In the S phase, the total DNA is doubling. So we write it as 4n. In gap 2, the cell containing the doubled DNA amount is staying, and then it is entering mitosis. At the start of mitosis, the DNA is doubled. But at the end of mitosis, you can see we are getting two daughter cells having again a diploid set of chromosome. So from the DNA point of view, if you see, with the start of mitosis, it is having a double DNA content. And as we move towards the end of mitosis, its DNA content has decreased. Interestingly, if we look into the S phase, at the start of S phase, we have a diploid set of DNA content. And at the end of S phase, we have the double DNA content. So there is an increase. Externally, if we see the DNA, if you want to understand in which phase the cell is, then simply by assaying the DNA content of a cell, it can be assumed that at which stage the cell is. So once more, we have a diploid set, 2n amount of chromosome here. At the start of S phase is 2n amount of chromosome. The DNA doubles, so it's 4n. So if we see that the amount of DNA is increasing experimentally, we can assume safely that the cell is in S phase, so the DNA is doubling. If we see that the doubled DNA amount is remaining constant for a certain amount of time, we can assume that the cell is in gap 2 phase. If we see that the DNA amount is decreasing as the cell, as time is moving, with time the DNA amount is decreasing, we have to assume that the cell is in mitosis because it starts with 4n and ultimately yields two daughter cells, 2n, 2n. And if we see that a cell is sitting at a doubled DNA content for, the cell is sitting for quite some time at a diploid DNA content, then we can safely say that the cell is in G1 phase. So simply by studying or assaying experimentally the amount of DNA present in a cellular system, we can assess at what stage the cell is in the cell cycle. So from this basic concept, we understood one thing, that just by assaying the amount of DNA at a particular time in a given cell, which is dividing, a batch of cells which is dividing, we can have a rough idea as to in which stage the cell is. So again, once more summarizing, the cell has a mitotic stage which takes the least of all the time. The maximum is G1, which takes 6 to 12 hours. Then we have the S phase. I'm talking about hours with respect to a human cell division, which takes for roughly 6 to 8 hours. Then we have a gap 2 phase, that is 3 to 4 hours. And then we have a mitosis 1 hour. This comprises the cell cycle of a cell. There is one phase which is not present in this picture, but is transiently present if there is any problem in the cell. So let me talk about that phase also. That phase is known as G0. So in this particular picture, you are not seeing the existence of gap 0 phase at any stage. But this gap 0 phase exists in the G1. Why does it exist? The question is that when a cell comes out from mitosis, it has just now finished a process of karyokinosis, that is nuclear division. The cell, the proteins in G1 needs to check whether the division went right or not, whether there was any wrong, anything missing in that cellular division. So checking is usually done in gap 1. And during this checking, if some abnormality is observed, if a DNA breakage is observed, any kind of abnormality is observed, then a question comes in front of the cell that should I proceed in this defective format or should I halt? Now, you see, our body is a evolutionary miracle. So the moment the cell has a cellular damage at the DNA level within itself, the cell realizes that if it proceeds in this manner, then it will be producing a progeny of defective cells. So it needs to halt, it needs to think. So as we saw when we talked about gap 1, S, gap 2 and M, there is no stage where the cell could halt. So whenever there is a defect of some sort in the cell, and that is identified in gap 1, the cell is prevented from proceeding further. It is halted, it is stopped, and it transiently halts in a stage in the cell cycle known as gap 0. It halts there, it waits, thorough checking is done, or it waits for some of the vital stimuli that are required for this particular cell to proceed further. So we should always remember that when a cell enters a G0 phase, there are two criteria, basic criteria, for which only a cell can enter a G0 phase. The first criteria is the nutrient availability, to check that whether food is available for the cell or not. If nutrient is not available, then a normal cell can enter a G0 phase. It becomes quiet and it waits for the availability of nutrients. The moment nutrients are again available, it comes out from G0 and re-enters G1. So that is one point by which we saw that a cell can enter from a G1 to the transiently present G0 phase. The other point for entering G0 is that, as I was discussing, that if there is a defect in the cell at the genomic level, so if there is a defect in the DNA, then the cell needs to be repaired. In that case, the sentinels or the guardians present in G1, they do not allow the cell to pass. The cell enters the G0. In the G0 phase, the cell is thoroughly checked. If the cell can be repaired, then it is returned from G0 to G1 and it can proceed for S. If the cell cannot be repaired, then the cell is instructed to die, a programmed cell death. So in nutshell, thus we have the 4 stages of cell cycle: G1, S, G2, M. S is the stage of cell cycle where we see synthesis of DNA, and M is that stage of cell cycle where we see division of that DNA. And we have a transient stage G0 where normal cells can enter G0 if there is no nutrient availability, or a normal cell which has some kind of defect can enter G0 for repair if no repair is seen, then the cell undergoes apoptosis. So that was in nutshell about the cellular life of a cell. Now let's look into this picture. I have in details discussed. So this is the stages I very clearly discussed with you till now, that a cell passes through M, G1S, G2, and the number of hours a cell stays in each of the stage. And as I was saying, when I discussed you the amount of DNA present in the cell, and we can identify based on the DNA content that in which stage the cell is, you can see this is the number of cells and this is the amount. So when we move from left to right, the amount of DNA is increasing, and you can see that 2n means G1. And I told you that as we move from 2n to 4n, if we have a increasing amount of DNA content, we have to understand that the cell is in S phase. So the cell is in S phase. And then again, if we have a constant 4n content, then we have to assume that the cell is in G2 phase. And again, which is not shown anymore in the figure, if we have a decreasing content from 4n to 2n, we can assume that the cell is in M phase. So just by assaying, analyzing the DNA content, for a batch of cells, we can identify in which stage of the cell cycle the cell is in. Now coming to this particular picture, because this particular picture is showing you this stage, the G0. In fact, G0 was shown here also. It's terminally differentiated cells withdraw from cell cycle indefinitely. So if the need arises only, then they can come back into the cell cycle, but otherwise terminally differentiated cells withdraw from cell cycle indefinitely, and this can be provided by the example of fibroblast cells of our body, the skin cells of our body. And as I was talking about the different stages of cell cycle, I told you that there is this one point which is very important in gap 1. This checkpoint is very, very important because this checkpoint ensures that the cell has defined amount of nutrients available. It also ensures that there is nothing wrong with the chromosome or the cell that has come out from mitosis. And this checkpoint is famously known as restriction point, and in East, it is known as the start point. It is influenced by these factors. So there's the presence of the growth factors, there's the presence of the nutrients, cell size is important, but this is in case of East. And obviously, and one of the most important thing is to check DNA damage. As one of you asked, and as this picture also shows, there are some more checkpoints available here. One is at the transition of G to M. This checks if there is any DNA damage or anything wrong went in DNA replication. And there is another checkpoint in the mitosis itself, which actually controls the metaphase anaphase transition. That is very vital. If you remember, in the metaphase anaphase transition, what happens in metaphase? The cells are lined, and then in anaphase there is a cellular pull and the chromosomes move to the two poles of a cell. So this lining and pulling of the chromosome is very vital. So there is again a DNA damage checkpoint which checks whether this is going on properly or not. And these are some beautiful pictures I just wanted to show you so you can have an idea that how the cell looks. You can see the G1 phase, and you can see the doubling of the DNA or the sister chromatids. And that is how the cell enters G2. So as I was saying, we are having 4 unreplicated chromosomes. Here also we have 4 unreplicated chromosomes, but the amount of DNA has increased. So when we compare now I am coming to this concept. When I am comparing the chromosome number and the DNA content, there is a difference. If I look into G1 of the parent cell, we are seeing 4 unreplicated chromosomes, right? During replication, each chromosome is doubling. In that manner, I had said it was 2n to 4n. Now, to be more precise on what is actually happening, is that the DNA content has totally doubled, but the 2 chromatids, as you can see, are attached at the centromeric region. So if I count the number of chromosomes, the chromosome is still 4n. If I do the count, it is 4n. The difference between G1 and G2 in this particular picture, as you can see, is the amount of DNA present. Now, if you count the chromatids, it is becoming 8 in G2. But if we count the chromosome once more, G1, 4 chromosomes, G2, 4 chromosomes, each chromosome is now having 2 sister chromatids. So from the chromosome point of view, we are having 4, 4. From DNA point of view, C represents DNA content. We are having 2C, 4C. So now, again, we are having 4 replicated chromosome. The difference is, this is 4 unreplicated chromosome. This is 4 replicated chromosome. So this is the difference occurring with respect to chromosome. We are having in G1 4 unreplicated chromosome, in G2 4 replicated chromosome. We have a 4C content of DNA. In G1, we have a 8C content of DNA in G2. So the DNA content has doubled. Now the cell has entered mitosis. At the start, this is the start of mitosis, M phase. The replicated chromosomes condense. And then during mitosis, as you can see, the chromatids separate and the 2 daughter cells are formed by cytokinesis. This is another beautiful picture. So if you look into this picture from the point of view of interphase, I just missed out that particular word interphase. So just let me get back to interphase. When we're discussing this stages of cell cycle in details, all these stages that includes G1, S and G2 is together known as interphase. And as you can understand, this is one of the most active stage of the cell. Though many of us refer to as a resting stage, but we have realized that the interphase is not actually the resting stage. The cell is doing a lot of work during the interphase. So let's get back to that particular slide again once more. So this is how the cell looks. So we are having this interphase. During this interphase, after chromosome replication, each chromosome is composed of the 2 sister chromatids. Centrosomes have also replicated. This is the prophase. Early spindle apparatus is forming. You have all idea about this. This is the prometaphase. So you can see the formation of the gradual formation of the mitotic spindles. You can see metaphase, the alignment of the chromosomes on the mitotic. Alignment of the chromosomes at in the middle of the cell. And this is anaphase. So these 2 are again vital because this alignment is important, and then the pull on the 2 sides. And hence there is a DNA, there is a checkpoint, damage checkpoint here, which actually controls this phenomena. We'll go over them in very brief. And then we have this anaphase, and then telophase, and finally the cytoplasm divides. So that is basically about mitosis. And as I was talking to you about the difference between the chromosome number and the DNA content. So what we can say, chromosome 2n you can see that this is M. Chromosomes 4N, that's 4C DNA content. Chromosome 2N, 2N, that has been represented as 2N. Let us look into this picture. This is centromere, chromosomes 2N, DNA content 4C. That was the concept I just now told you. That even though we see the doubling of the chromosomes, what happens? The chromosomes remains attached in the centromeric region. And thus, if we started with 4 unreplicated chromosome in G1 phase, after S phase in G2, we get 4 chromosomes, but these have now replicated and the sister chromatid is attached in the centromeric region. So even though we started with 2N number of chromosomes and 2C DNA content early S phase, you can see the chromosome number is remaining constant, it's 2N. But the difference is now we are having replicated chromosomes. But that is one part. And as I said, we started with 2C DNA content, and due to DNA replication, we will now say that the DNA content has now become 4C. Okay. When we are entering M phase or mitosis, we start with 2N chromosome and 4C DNA content. At one point in mitosis, when there is this metaphase and this anaphase pull, if you remember, we are at a given point having the chromatids separate. So at this point, you see the chromatids have become separate. So the chromosome number has transiently doubled. So now we can say that there are 8 chromosomes. So that is what it has written. For that transient period, the chromosome number doubles. And then there is this division. So again, we are having 2N number of chromosomes and 2C DNA content. So this is the relationship of the chromosome and the DNA content during the cell cycle. We'll stop with this.